Okay, hello everybody. I want to introduce the second speaker today, as you just heard. The second speaker will be Jo McEntyre. She joined the European Bioinformatics Institute, that's the site that EMBL has in the UK, as a team leader in 2009. And since 2019, she's the Associate Director for Services and the Head of Literature Services there. She co-chairs, together with Wolfgang Huber, the EMBL Working Group on Open Science, and she's also responsible for Europe's PubMed Central, the PMC. This is the database for full-text life science research articles. I'm sure many of you have used this. And this is the one that's based at the European Bioinformatics Institute, and it's partnered with the US PubMed Central. The primary focus of Jo's work is on the integration of open science literature with open science data, and her work aims to build infrastructures that support the development of new ways to access and discover useful information in research. And particularly, she's an expert in the areas of text and data mining, publication workflows, and scientific credit systems. Before she joined EMBL, Jo was a scientist at the National Center for Biotechnology Information, NCBI, at the NIH in the US. There she worked on various literature-related resources and website usability. And before that, she was an editor at the journal Trends in Biochemical Sciences. And her PhD was in the field of plant cell biotechnology. Her talk today is going to be entitled EMBL Open Science Culture. So welcome, Jo.
Thank you for the opportunity to talk to you as part of the Sister Institute seminar series about EMBL Open Science Culture. So EMBL was established in 1973 as an international organization. And looking through the establishing agreement, one of the clauses states specifically that the results of the experimental and theoretical work of the laboratory shall be published or otherwise made generally available. And I don't know what the people who wrote this agreement had in mind in 1973, but perhaps today this might be translated as "will practice open access or open science." So how is EMBL delivering on this part of the agreement today, nearly 50 years on? Well, just to add a bit of context, in the early 1980s, the state-of-the-art technique was DNA sequencing. And the way to make the DNA sequence available that you'd so carefully, or the RNA sequence available that you so carefully done, was to actually write the sequence out in the papers where the sequence was described. Of course, nowadays this would be lunacy. And in fact, the people at the time had a lot of foresight in realizing that, increasingly as the technologies became more widely available, that many sequences are becoming available, and we should do something better than writing out sequences in papers. Obviously, the human genome would never fit in an abstract. So the question of the need for a data bank in which to contain all these sequences so that they could be shared for reuse and reanalysis emerged, and EMBL decided to act. And what seems quite obvious these days as a sequence data record was not so obvious at the time. And so if someone had to think about what kinds of fields would accompany the sequence data, and here you can see a draft from Graham Cameron, I think, who sort of launched the nucleotide sequence data library, as to what those fields might be. And at the top you can see one of the most important ones here is it needs to have an accession number. This is a way to uniquely identify the data record that you're talking about. A date, which has still remained, and FT stands for feature table, where the sequence itself and other features are described. So a little further on from this, obviously realized this was a global effort, not just a European effort. And so, after several meetings with the NIH and later the Japan, the International Nucleotide Sequence Database Collaboration was derived. And this is an international organization that stressed the need for biological information infrastructure. So not just a couple of databases here and there, but a proper way to support this kind of work and requirement. As the world became more serious about sequence databases, and we realized, of course, that many sequences of great length would be produced, the journal stepped up to the plate to start requiring that instead of writing out the sequence in the paper, that you would put the sequence in the database, the EMBL data library, get an accession number, and then cite that accession number in the paper as proof that you deposited the data for all to scrutinize. And Nucleic Acids Research was the first journal to do this, but many other journals quickly followed, and now it is basically common practice to cite the data in the paper and deposit the data in the community database. So thinking about, with that as a background, thinking about open science, and this is a modified quote from John Clairbout, who was a geophysicist in Stanford. And he basically states very eloquently that a scientific publication is not the scholarship itself, it is merely advertising of the scholarship, and that the actual scholarship is the complete set of reagents, data, and analysis steps or code, which generated the figures and the tables. And so what we have in this iceberg analogy, where what we can see, which is the paper, hides a morass underneath that is very hard to find even now. And here's the good ship EMBL navigating these waters, as any other institute is at the moment, I would expect. And what we must try and do is float the iceberg so less of it is undercover, as well as remove barriers potentially to accessing that information. And that's what we're tasked with trying to do. So what is open science? And we've shamelessly paraphrased from Wikipedia here. We're going to make open science, in our definition, is making the results of scientific research, including publications, data, samples, and software, accessible to all levels of an inquiring society, whether those people be amateur or professional. And to make those data provide transparency and accessible knowledge so that it can be shared and developed through collaborative networks. EMBL, so from these beginnings of the European nucleotide data library, we now have one of the units of EMBL, the European Bioinformatics Institute, where we host many molecular biology databases. And these databases come in two types. We have deposition databases, where experimental data from a given community, for example, proteomics or expression data, are deposited in community databases so that they can be analyzed and reused. And on top of that, we had added value databases where we make some view of those experimental data that makes it more accessible for most people. And a good example of that is genome assembly, genome assemblies that are based on the raw data in the European Nucleotide Archive. And because we do a lot of collaborating to generate these databases, we often do a lot of work on standard development for data, such as the metadata you saw in Graham's sketch earlier. And we try and link where possible to the literature. And one of our operational principles is that all data are free to access at the point of access, and they're free to download and reuse by anybody in the world. In the tradition of databases for different communities, and supporting the cutting edge of science, one of the most fastest growing technologies at the moment is imaging technologies. And EMBL has responded to this in two ways. The first way is by building an imaging center, which is based at Heidelberg, which will provide services around image-based interrogation across scales for the research community. So provision for everything from molecule scale to tissues and organs. And this is really built on the fruits of EMBL experience and what a part of our remit, which is technology development. And it's not just these microscopes are so cutting edge that they're developed with industry partners as well as other developers. And this is not the only resource. This is part of a Eurobioimaging network. And so, in general, there's going to be a vast amount of imaging data being produced, petabytes and petabytes, in the next few years. So to respond to that, the EBI recently launched the Bioimage Archive. This is a new database. It was only released last year, and it's open for submissions. And it basically provides the means to share data that is linked to research papers. And how we see this working is that it'll be the sort of back end of an ecosystem of different community databases, so that those can have specialist annotations and specialist metadata, and it will be powered by the BIA. And it'll provide the foundation for much AI and analysis methods development, as you can be able to search across data sets from a number of different resources. Given the current circumstances, the other recent thing that EBI has done is release the COVID-19 data portal, which is really a means to share COVID-19 related data in a coherent sense. And this includes new viral sequence data from data hubs across Europe, as well as host or human data, which will be provided by Federated European Genome Phenome Archives. And this portal basically puts this kind of data, this new rapidly increasing data, together with other research information on structures, proteins, expression in the literature, and so on. So moving from what the EMBL does via the EBI largely for the wider community, what is EMBL doing in its own backyard? And we've recently set up an EMBL Open Science Working Group to encourage open science best practices at EMBL. What our key task is to revise internal policies, which is our internal rule book, which will describe how we expect the results of research to be made available. And then once we've sort of agreed on that, then we can define processes to support people in doing that. We can identify how we can get people to do it, and also some methods probably to describe how we can measure the outcomes. And so why do we want to do this? Well, as in our establishing agreement, open science is always the future. And it's not just that, but the funders are increasingly requiring us to do it and report on it. And finally, EMBL is visible in the community, in particular as its role as a training institution, and we want to make sure that those that touch our training programs in various different ways are well grounded in how to share their research in an open way. So we're focusing on publications, data, and software, and the path for open access publications is fairly well trodden. We have a database run at the EBI called Europe PMC, which is a database of full text research articles, both peer-reviewed ones and increasingly preprints. And at the EMBL, we essentially use this as our open access repository. It's also used and supported by several funders of biomedical research, including WHO, the ERC, and Wellcome Trust. But for software and data, it wasn't quite so clear what to do. And we realized when we were going to make a policy, the first thing you have to do is be clear about what it is that you're talking about. And for software, we have decided that there's only three types really. This is after a lot of discussion. That basically the software that supports the results in a research paper. Then there's software that supports our services. And then there's sort of an in-between category, which is software that often emerges from research groups that ends up being more widely used as a tool or a method. And all of these types, we want to make them open to support transparency, improve the quality of research, and make more accessible for peer review. And in some cases, such as the general purpose tools and methods, we expect those to also be reusable. For data, we have decided that, well, we are pushing towards all data behind publications to be published, not just cherry-picked pieces of data. We are when there's a community database, we expect that community database to be used as a priority, And then, generic databases may be used for other kinds of data sets, such as supplemental data files, what would normally be considered files appended to a paper. And then what we're very seriously thinking about is requiring data management plans for all projects, because we recognize that sometimes it's not 100% clear what data to publish, how refined it should be, how much there should be made available. And all this will require training and dedicated human capacity, as well as various tools. Which brings me to DORA, which is an agreement to try to stop using journal-based metrics solely for research assessment. And here we're really talking about the impact factor, where people will say, something like, oh, that person has a Nature paper, so they must be good. So how can we get away from judging research by the venue in which it's published? We really do have to look at the research itself. And this doesn't necessarily mean reading every research paper in someone's CV, but rather I think it will mean making more opportunities from our technical abilities that we have these days, as well as looking at alternative research outputs and inventing new indicators of quality and new metrics around those. And this is where I think EMBL can really help, is in particular through its data resources, provide a venue on which to base new ways to assess research. Which brings me to Jeremy Farrar, who thinks that we should reimagine how we do research, and as a community, how we can change our approach so that we can encourage diversity and improve the quality of research by doing things in a more open manner. And I really hope that through our EMBL programs of services and so on, that we can contribute to that, both as an institute in the research that we generate, as well as through the training and the services that we run. And it remains me just to thank our Open Science Working Group, who put a lot of thought and a lot of effort into thinking about how we progress this at EMBL. And I'll take any questions. Thank you.
Thank you, Joe. That was a really excellent talk, and I'm glad that we were able to hear the end of it and that you were able to come back and join us for some live Q&A. So my first question to you is that obviously open science is a very hot topic right now, and the argument in favor of open science is very compelling. But in your opinion, do you see any disadvantages or shortcomings to the idea of open science?
Well, I have to say, I don't. I mean, I think it's going to be, I think it's going to take quite a lot of work to do and quite a lot of support to do, and a change in the way we think about things. So I don't see any negatives, except that it's, we're going to have to really make a big effort and have support to do it. Maybe someone would disagree with me.
All right, so we have some good audience questions here, and I'd like to go to Jim Cox. Jim, would you like to ask your question?
Yeah, sure. Hi, Joe. When using animal models, a big component of the iceberg below the water is the procedures for housing and care of the animal model. How is EMBL dealing with the tracking the variations of animal housing and care, which can have a profound impact on the result of the studies that involve animal models?
That's an excellent question. So you're talking about physical samples and the ways in which those physical samples have been kept, if I could generalize the question.
Yeah.
Well, I might turn to my colleague Cornelius, who has more experience in running model animal facilities. But I think this is a really good and important question. It's not just mouse models and so on, but also physical samples and what temperature the fridge was at and all that kind of thing. So I think really we need better databases that will help describe that, and I believe there are a few that are emerging, but yeah, that's a pretty difficult, physical samples are a pretty difficult problem.
Joe, we have another question from Wyatt Korf. Wyatt, are you there?
I am, thank you. Hi, Joe. Thanks for the talk. I'm wondering, following up on your giving credit for the work that was produced, and I love that you guys are promoting ORCID. Have you guys investigated use of persistent identifiers for the research group or for the teams, something like the RAID initiative?
I know of the RAID initiative. It's like a project ID, and yeah, I mean, we could definitely think about doing that. The two things you really need to have wide uptake there is like a infrastructure to support it, and then the reasons for uptake. And so I think it would certainly be better to have that kind of thing, if rather than all these consortium names and things like that. It would be difficult, but it's how to drive the uptake that's it's not really the technical part that's the problem. There's always there's technical ways to do things. It's a question of the behavior and the uptake.
Yeah, I think it's the real question is, how do we incentivize the collaborative work, and is giving credit to an individual versus the entity really the best use of that credit model?
I agree. It's a good question. I mean, certainly the physicists have had a better approach to team science for a long time than the biologists have. But I do think we have an increasing number of consortium authors in the literature, so it suggests that we are somewhat improving, and there are ways also to now describe the role of different authors on the paper. I mean, I think it's very difficult when the only way we get credit is to be an author on the paper, and therefore the people on papers that haven't really authored the paper, they might have contributed in some way, but they didn't write the paper. And there's a lot of work to do here to change the way that people are rewarded for their good work, and which mechanisms we have to hand that allow those assessments to be made. And we just have to, it's a massive task. Because I mean, sorry.
Go ahead, just finish, go ahead.
Oh, I was going to say, even if, if we consider EMBL changes the way it does research assessment, we still have to train young researchers who might be going off to get, jobs in other parts of the world or in other institutes that are still looking for, nature, science and cell papers. So, it will require, some leadership, but also then changing culture in many different institutes. So it's a pretty tough call. You've just got to find the things you can do and do them.
Thanks, Joe. We have a few other good questions. So if you don't mind answering just a couple of more for the people that are out there, that would be awesome. Is that okay?
Sure.
We have one from Mike Parham. Mike, are you there?
I'm here.
Go ahead, Mike.
So I was wondering, in addition to what you're doing at EMBL, which is providing a lot of infrastructure and expertise to support all these open science initiatives, are there other institutions that you look to benchmark yourself, or others that you just think they're doing things well?
That's a very good question, and I'd really like to do that. So there's two parts. There's the outward facing research facilities. And when we have sort of sister institutes there, really, we collaborate widely with data resources across the world to build those, and researchers across the world to build those data resources. And I think we do benchmark with those, and probably benchmark, but also collaborate. If you're talking about EMBL as an institute and how we're doing with our research outputs, that would be very interesting. I don't know how to do that, but we certainly could look to similar institutes and talk with them about what they're doing and their approaches. I think that'd be a very good idea once we've advanced our own open science thinking a little further.
We have another question from Brigitte Kerber. Brigitte, are you there? Would you like to ask your question?
Yeah, just Joe, many thanks. This was an excellent talk and overview of what is going on at EMBL. Do you have a suggestion as for alternatives to using the journal impact factor as a measurement for the quality of a piece of research?
Well, yes. Again, it's a question of uptake. So, one thing you look at is the citation of the paper itself, not just the impact factor of the journal. So if you want to stick in the land of papers and citations, you can do that. But I think we really do have to start looking at other forms of impact. and, timescales of impact. So, open software, people who develop tools that really change the world and change the way that many people can do their science, has a tremendous impact, and it should go way beyond the publication of a single paper, for example. Same with some data resources too.
Let's see, we have another question from Bodo Stern. Bodo, are you there?
Yes, can you hear me?
Yep, go ahead.
Okay, hi, thank you, Joanna, that was a great talk. You mentioned that in your with your data that scientists wouldn't need to share the whole data set, they wouldn't be able to cherry pick, and that seems great. But so my question is, for clinical trials, you can imagine that every data that are part of this trial need to be published. But with basic science, there are a lot of data sets that are not even worth publishing, and that will be up to the authors to decide what should be, what should go in. Is that how you see it as well, that there's still this decision of the author what data they should share, or is it once they have a data management plan that pretty much the data they generate should be public?
Yeah, so I think with an experiment that delivers, for example, a lot of related images, the cherry picking part refers to the archiving of the whole data set. And so the paper often will talk about, sequencing papers as well. The paper will often talk about a few parts of the most interesting bits to that author at that time of the work they've done. So it's really about, depositing the whole genome, not just the bits of the genome you're interested in. That's that bit. The bit I totally get, and I think it's really different. We talked about it for a long time. It's really difficult. Like, what do you mean all the data? Do you mean all the raw data? Do you mean the process data? Do you mean my data that I know is not quite right, or it's not even useful, just creating noise? And so I think for that we do need a bit of data stewardship and support for decision making. Because I understand, talking to my colleagues, that it's they're quite uncomfortable sometimes in thinking about what they want to publish as much as possible, but it's a difficult decision to make as to how much to publish sometimes. So I think we need, the first part would be a data management plan. This is what we're thinking. Then at least what might be produced in the future, and then you can start to think about what the best way to deal with that would be. And there might be a gradient of what you publish. There could be masses of it, the great morass of data on tape. There could be layers of data, so raw data, process data. I think we need expertise to help us decide that. I don't think it should be it should be driven by individual opinion, but we should have an institutional view on what the best practice is. And without knowing what all the data are, it's quite difficult to do. Therefore, the data management plan idea is a sort of hedge on that we can at least begin to think about it in a more structured way.
Thank you very much. Okay. Very helpful.
Thanks, Joe. I think we need to end it here, but I do want to remind everyone that they could head over to Slack. The link is in the chat box. If you want to ask the speakers any further questions. I know that we weren't able to get to all of them today, but I just want to close by thanking very much again our two speakers today, Nassos and Joe. They were great talks and great Q&A and conversation afterwards. I want to thank the audience for joining us today, and we really appreciate it. And I want to encourage you to come back next Wednesday on August 5 to hear from the Institute of Infectious Disease and Molecular Medicine at the University of Cape Town in South Africa. We will hear from Valerie Mizrahi and Anastasia Koch. So I look forward to seeing everybody next week. Thank you.